CirRNAPL, a web server for the identification of circRNA based on extreme learning machine Mengting UA, 1, Jun Zhang B, 1, Yanjuan Li C, Cankun Wangd, Zhaoqian Lu D, Hui Ding E, Quan Zhou A, Qin Mad, Institute of Fundamental and Frontier Sciences, University of Electronic Science and Technology of China, Chengdu 610054, China B Rehabilitation Department, Heilongjiang Province Land Reclamation Headquarters General Hospital, Harbin, China C School of Information and Computer Engineering, Northeast Forestry University, Harbin 150,040, China D Department of Biomedical Informatics, Ohio State University, Columbus, Ohio, USAE Center for Informational Biology, University of Electronic Science and Technology of China, Chengdu 610054, China Article Info Article History, received November 16, 2019, received in revised form March 29, 2020, accepted March 29, 2020, available online April 2, 2020. Keywords: circRNA, extreme learning machine particle swarm optimization algorithm identification expression level abstract. circRNA, circRNA, plays an important role in the development of diseases, and it provides a novel idea for drug development. Accurate identification of circRNAs is important for a deeper understanding of their functions. In this study, we developed a new classifier, CirRNAPL, which extracts the features of nucleic acid composition, structure of the circRNA sequence, and optimizes the extreme learning machine based on the particle swarm optimization algorithm. We compared CirRNAPL with existing methods, including BLAST, on 3 datasets and found CirRNAPL significantly improved the identification accuracy for the 3 datasets, with accuracies of 0.815, 0.802, and 0.782, respectively. Additionally, we performed sequence alignment on 564 sequences of the independent detection set of the 3rd dataset and analyzed the expression level of circRNAs. Results showed the expression level of the sequence is positively correlated with the abundance. A user-friendly CirRNAPL web server is freely available at http://server.malab.cn/cirrnapl/. 2020. The authors. Published by Elsevier BV on behalf of Research Network of Computational and Structural Biotechnology. This is an open access article under the CC by NC/ND license, http://creativecommons. org/licenses/by-NC/ND/4.0/. 1. Introduction. circRNA, circRNA, is a newly identified RNA type that differs from conventional linear RNA in humans. It is a non-coding RNA molecule without having a 5-end cap or a 3-end tail, instead, forming a circular structure, 1,2, Figure 1. circRNA was first discovered in 1969 by Diener, while researching potato spindle tuber disease. 3. Electron microscopy revealed the formation of such closed-loop RNA, also known as a viroid. The subsequent emergence of high-throughput sequencing techniques, RNA-seq, enabled improved sequencing of circRNAs of various species, and many circRNAs have now been identified, 4,5. To date, more than 10,000 different circRNAs have been successfully identified from fruit flies and worms to mice and humans, 6,7. Against background researches and applications of circRNA, a series of databases have been built, such as CircBase, 8, CircNet, 9, Circ2 Traits, 10, TSCD, 11, circRNAb, 12, and CircInteractome, 13. Studying the structure and function of circRNAs, researchers have revealed their importance in the pathogenesis of arteriosclerosis, nervous system disorders, diabetes, and tumors. https://doi.org/10.1016/j.csbj.2020.03. 028-2001-0370/2020. The authors. Published by Elsevier BV on behalf of Research Network of Computational and Structural Biotechnology. This is an open access article under the CC by NC/ND license, http://creativecommons.org/licenses/by-NC/ND/4.0/. Abbreviations: circRNA, circular RNA, RF, random forest, PSO, particle swarm optimization algorithm, uncRNAs, long non-coding RNAs, PCGs, protein coding genes, DAC, dinucleotide-based autocovariants, DCC, dinucleotide-based cross-covariance, DAC, dinucleotide-based auto-cross covariance, MAC, Moran autocorrelation, GAC, Geary autocorrelation, NBAC, normalized Morrow-Brodo autocorrelation, PCS-DNC general parallel correlation pseudo-dinucleotide composition, SCS-DNC general series correlation pseudo-dinucleotide composition, triplet, local structure sequence triplet element, SSC, pseudostructure status composition, STPC, pseudo-distance structure status pair composition, ELM, extreme learning machine, RBF, radial basis function, CNN, convolutional neural networks, SVM, support vector machine, SE, sensitivity, SP, specificity, ACC, accuracy, MCC, Matthews correlation coefficient, MRMD, maximum relevance maximum distance. Corresponding authors at Institute of Fundamental and Frontier Sciences, University of Electronic Science and Technology of China, Chengdu 610054, China, Huzhou. Department of Biomedical Informatics, Ohio State University, Columbus, Ohio, USA, Q. Ma. Email addresses, socanetlab.net, qzo, chin.ma@azumc.edu, Q. Ma. When these authors contributed equally to this study, Computational and Structural Biotechnology Journal 18, 2020, 834-842 Journal homepage, www.elsevier.com/locate/CSBJ, 14-16. The unique endogenous characteristics of circRNAs enable their value as biomarkers, providing a new approach for drug development, 6,17,18, and a new direction for the evolution of life. Xu used Qualcomm sequencing to detect the expression profiles of 3 wheat samples, LH9, XN979, and YN29, and identified 33 differentially expressed circRNA, 19. Yi used published RNA-seq data to perform genome-wide identification of circRNA in rice and Arabidopsis. Based on this, they compared the characteristics of plant and animal circRNA, 20. Moreover, Mengting analyzed circRNA from humans, mice, and nematodes, and detected thousands of well-represented, stable circRNAs, demonstrating their regulatory capabilities, 21. Given this landscape, accurate detection and quantification of circRNAs are essential for a deeper understanding of their function. In addition to the abundance of RNA-seq data, a variety of algorithms for calculating and visualizing circRNA have recently been developed, 2,17,22. Li Chen developed the circExplorer software based on RNA-seq data of Arabidopsis and rice, providing a more comprehensive and sensitive circRNA prediction method for plants, 23. Song et al. Developed a computational pipeline, called Ouroboros, to detect circRNA in RNA-seq data. They successfully verified 24 circRNA from 27 randomly selected circRNA, 6. Additionally, based on RNA-seq, Danon developed a circRNA-seq method for reading circRNA in an unbiased, genome-wide manner. Moreover, they mapped the transcriptome of Salfatericus, 24. In another study, Yek et al. Used high-throughput sequencing of ribosomal deficient RNA and identified more than 25,000 loops in human fibroblasts containing non-collinear exons, reverse splicing, 25. Gao et al. Proposed a new algorithm, CirRNAPL, based on cross-shear signal, which uses a variety of filtering strategies to accurately detect circRNA from transcriptome data, 26. Zhang et al. Used non-polyadenylation and renaser-treated RNA-seq data from H9 human embryonic stem cells to predict reverse splicing junctions, and they systematically characterized circRNA using the newly developed pipeline circExplorer, 27. Furthermore, Bo et al. Used the exome capture RNA sequencing protocol to detect and characterize circRNA across greater than 2,000 cancer samples, establishing the most comprehensive catalog of circRNA species to date, Mi-OncoCirc, 28. Yu et al. Proposed the method, X, allowing for the redefinition and accurate and rapid identification of circRNA from single-ended and double-ended RNA-seq data, as well as their abundance, 29. Furthermore, Zhang et al. Sequenced Branchiostoma belcheri circRNA and identified 1859 circRNAs using the find_circ and CirRNAPL algorithms, 30. At present, many available recognition methods are based on RNA-seq data, And it is very important to use bioinformatics with direct circRNA sequence training to achieve more accurate recognition. In contrast to the tools that use RNA-seq data as input, Pan et al. Extracted the characteristics of the sequence and used the random forest (RF) algorithm to identify circRNAs, subsequently building the online identification server web circRNA. 31. Pre-circRNA classified circRNA and LungRNA based on a computational method of a multi-core learning framework with multiple functional training. 32. HELM used a hierarchical extreme learning machine algorithm with feature selection to extract the same features from other LungRNA and classify circular RNAs. 33. Mohammed Chaban proposed an end-to-end deep learning framework circDeep, which integrates shared representations across different modes and improves circRNA for classification. 34. Many studies for protein recognition, 35, and site detection, 36, have been performed based on machine learning, such as RF, 37, and artificial neural network, 38. By contrast, few studies focus on the identification of circRNAs. Therefore, there is a need for studies on how to use sequence information to achieve more accurate identification by using the characteristics of RNA sequences. We propose a new method that mainly uses the structural features of RNA and nucleotide composition to optimize the extreme learning machine, ELM, based on the particle swarm optimization algorithm, PSO. Based on these, a classification system was built to optimize the effect of circRNA identification. 2. Material and methods. In this study, we use the ELM method to identify circRNA. The flowchart of the identification framework is shown in Figure 2. 2.1. Dataset circRNAs are evolutionarily conserved among different species, and the detection of circular RNAs is important for further understanding the biological origin and purpose of circular RNAs. 39,40. The datasets that we used were presented in the literature of Pan et al. 31. Table 1. The literature downloaded 92,375 circRNA transcripts from circBase. After deleting transcripts of less than 200 nt in length and the overlapping circular RNA transcripts, 14,084 circRNA data were obtained. The database source of the 9,533 PCGs and 19,723 LungRNAs dataset is GenCode 19, in which overlapping circRNA sequences were removed from the PCG data. The third dataset includes 2082 circRNAs from stem cells expressed in H1-seq and the same number of identically derived circRNAs from other cells that were not expressed in H1-seq. And the circRNA versus LungRNA dataset also applied to the literature of Mohammed et al. 34, Pan et al. 32, and Chen et al. 33. The construction of the classifier used in this study involved tenfold cross-validation. To verify the classifier, independent training and test set verification was also performed. For the dataset of 14,084 circRNAs, the number used for the training set was 10,000, and that for the test set was 4,084. For the LungRNA dataset of 19,722, the number for the training set was 10,000, and that for the test set was 9,722. For the PCG training set of 9,533, the number for the training set was 8,000, and that for the test set was 1,533. Finally, for the two datasets of 2082 circRNAs of each of stem cells and other cells, the training set consisted of 1,800 circRNAs, and the test set consisted of the remaining 282 circRNAs. 2.2. Feature representation methods to identify circRNAs. 4 features of the sequenced data were extracted, including ribonucleic acid composition, autocorrelation, pseudo-ribonucleic acid composition, and predicted structure composition. 41. These 4 features comprised a total of 15 modes, wherein ribonucleic acid composition included K-mer, the param flanking intron left flanking intron right 5-3 linear RNA splicing circular RNA splicing spliced reads back spliced reads linear mRNA circRNA exonic figure 1. circRNA splicing structure. CircRNA is a new class of RNA that differs from traditional linear RNA. It does not have a 50 cap or a 30 tail and is not easily degraded by exonuclease. In humans, it is more stable than linear RNA. Most circRNAs are formed by exons, while a few are derived from intron fragments. Mu et al. Computational and Structural Biotechnology Journal 18, 2020, 834 to 842, 835 enter K is 2 and 3, mismatch, and subsequence. Autocorrelation included DAC, DCC, DAC, MAC, GAC, and MAC. Pseudonucleotide composition included general parallel correlation pseudodinucleotide composition, PCS-DNC general, and general series correlation pseudodinucleotide composition, SCST general, and predicted structure composition included local structure sequence triplet element, triplet, SSC pseudostructure status, and STPC. A total of 520 dimensional features were obtained through these 15 modes. Figure 3. 2.2.1. Ribonucleic acid composition. For the nucleic acid composition feature of RNA sequences, 3 features, including basic K-mer, mismatch, and subsequence, were used, of which K-mer, 42, is the simplest method for expressing RNA. Basic K-mer can then be used to calculate the frequency of occurrence of K-adjacent nucleic acids. Mismatch, 43,44, can calculate the frequency of K-length adjacent nucleic acids, which differ by up to M mismatches, M less than K. Subsequence is a method that allows discontinuous matching, 45. Suppose an RNA sequence R is as follows: R1 quarter R1 R2 R3 Rn eth1 thorn wherein R1 represents the first nucleic acid in R, R2 represents the second nuclear D2, I, XMI1 quarter 0 D4K, I. Eth2 thorn where DJ, I represents the number of occurrences of the JTH K-mer type in RNA sequence R, J1 quarter 1, 2, 4K, I1 quarter 0, 1, M. Lastly, the sequence feature vector obtained by using subsequences. Table 1. The details of the datasets. Datasets: positive data, negative data, circRNA versus PCG 14,084, circRNAs 9,533, PCGs, circRNA versus LNCRNA 14,084, circRNAs 19,722, LNCRNAs stem cell versus not 2082, circRNAs 2082, circRNAs. Figure 3. Feature expression method and feature dimension histogram. Figure 2. Flowchart of CirRNAPL. CirRNAPL identifies circRNA in 4 main steps: data, feature, classifier, and circRNA identification data. This involves dataset construction. According to the bed data file in the HG38 human genome, we write Python script files to extract the corresponding sequence data. Features: this involves the extraction of features. In this work, information such as the structural characteristics of the RNA sequence is used as the feature to be extracted, including 4 parts, with a total of 14 calculation models. Classification: this involves the construction of the classifier. Here, the extreme learning machine based on particle swarm optimization is used as a classification algorithm. The classifier CirRNAPL is constructed by a tenfold cross-validation method, and the final classification model is output. circRNA identification: the RNA sequence to be labeled is identified using the classifier CirRNAPL. 836 MU et al. Computational and Structural Biotechnology Journal 18, 2020, 834 to 842 liters 1 quarter XA1 US the 1 thorn, XA2 US the 2 thorn, XAJS AJF thorn 0 at 1 of when AJ is exact matching, SAJ 1 quarter 0, when AJ is noncontiguous matching, SAJ 1 quarter IJJ. Here, U represents the attenuation coefficient, U half 0, 1, J 1 quarter 1, 2, 4K. 2.2.2. Autocorrelation. For the autocorrelation feature of RNA sequences, dinucleotide-based autocovariance, DAC, 46, dinucleotide-based cross-covariance, DCC, dinucleotide-based autocross-covariance, DACC, 47, Moran autocorrelation, MAC, Geary autocorrelation, GAC, and normalized Morrow-Brodo autocorrelation, MBAC, are used, 48. DAC measures the correlation of the same physicochemical index between 2 dinucleotides along the sequence interval L. DCC measures the correlation of 2 different physicochemical indicators between 2 dinucleotides separated by L nucleic acids. MAC measures the correlation of the same property between 2 residues along with sequence interval L. GAC measures the correlation of the same properties between 2 residues of distance L. MBAC measures the correlation of the same properties between 2 residues at a distance L. 2.2.3. Pseudo-ribonucleic acid composition. For the pseudo-ribonucleic acid composition feature of RNA sequences, PCSDNC general and SCSDNC general are used. In the PCPNC general method, the user can select not only 22 built-in physical and chemical indicators, but also upload their own indicators to generate PCPNC general feature vectors. SCSD general is a variant of PCSDNC general 49. 2.2.4. Predicted structure composition. For the predicted structure composition feature of RNA sequences, triplet, pseudostructure status composition, SSC, and pseudo-distance structure status pair composition, SDPC, are used. Triplet, 24, is an early method based on RNA sequence structure information, and it shows better circRNA identification performance than other sequence-based methods, 50. 2.3. Extreme machine learning to identify circRNAs. The ELM is used here as the basic classification algorithm. It has also been applied to biometric identification, 40,51-55. ELM is a generalized single hidden layer feed-forward network. This algorithm randomly assigns input weights and hidden layer thresholds and directly calculates output layer weights by least squares. The entire learning process is completed once, and no iteration is required. Therefore, the algorithm learns rapidly. 2.4. Particle swarm optimization algorithm. Particle swarm optimization, PSO, is often used to neural network optimization due to its simple rules, rapid convergence, less parameter adjustability, and strong ability for search, 56,57. PSO is based on the behavior of group foraging, where the particles are the solution to be optimized, 58. PSO is used to optimize the input weight and hidden layer deviation of ELMs, which can improve the generalizability of the methods. The particle swarm ELM algorithm relies on fewer hidden layer nodes to achieve higher precision. 2.5. ELM of PSO optimization. The kernel function of the extreme learning machine has a significant influence on the performance of the algorithm. The kernel parameter R and penalty coefficient C in the kernel function have an important impact on the performance of the ELM. R affects the scope of the kernel function, and C affects the stability of the model. The paper used PSO to optimize the parameters R and C. The search space of the PSO corresponds to the parameters of the ELM. The position of the particles represents the parameter value, and the accuracy is used as a fitness function of PSO. The steps of the PSO optimization ELM are as follows. 1. Initialization. The number of iterations and the overall size were set to 50 and 50, respectively. A particle population was randomly generated. Each particle in the population consists of a set of R and C. 2. Calculation of fitness function value. The fitness function value of the PSO algorithm is the accuracy of the ELM. 3. Updating the best position and particle position of the population, as well as the speed and position of the particles according to the formula. 4. And 5. 58. Vith thorn 1 thorn 1 quarter x vith thorn C1 repist, I pith. Thorn C2 R2 half gist pith eth 4 thorn pith thorn 1 quarter pith thorn vith thorn 1 thorn eth 5 thorn where epith and vith are the position and velocity of the TTH iteration of particle, respectively. Pist, I is the optimal solution for particle I, gist is the optimal solution for the population, x is the weight. C1 and C2 are the acceleration factors, R1 and R2 are random numbers between 0 and 1, T is the number of iterations. 4. Checking termination conditions. If the maximum fitness value or the maximum number of iterations is reached, go to step 5. Otherwise, return to step 2. 5. Obtaining the optimal ELM parameter with the largest fitness value. In this way, the parameters of the ELM model can be obtained, and the circRNA is identified using the optimized ELM. 2.6. Performance measurement to evaluate the identification effect of the constructed classifier. 4 commonly used indicators were selected here: SE, sensitivity, SP, specificity, ACC, accuracy, and MCC, Matthews correlation coefficient. SE indicates the rate of correct prediction of positive sequences. SP indicates the rate of correct prediction of the counterexample. ACC indicates the correct rate of classification. MCC indicates the reliability of the classifier, which can reflect the prediction ability more fairly. A larger MCC reflects better reliability. SE 1 quarter TP TP Thorn FN eth 6 Thorn SP 1 quarter TN TN Thorn FP eth 7 Thorn ACC 1 quarter TN Thorn TP TN Thorn FP Thorn TP Thorn FN eth 8 Thorn MCC 1 quarter TP TN FP FN TP Thorn FP TP Thorn FN TN Thorn FP TN Thorn FN PF 9 Thorn here. TP indicates the number of circRNAs predicted correctly. FP indicates the number of non-circRNAs predicted correctly. TN Indi MU et al. Computational and Structural Biotechnology Journal 18, 2020, 834 to 842, 837 indicates the number of incorrectly predicted circRNAs. FN indicates the number of incorrectly predicted non-circRNAs. 3. Results 3.1. Feature importance analysis taking into account the efficiency of the classifier operation. MRMD, maximum relevance maximum distance, was used for feature selection and the output of feature score sorting results, 59. We analyzed the top 20 features based on the output feature scores, figure 4a, and get the 20-dimensional feature set obtained by feature extraction. On the circRNA versus PCG dataset, the first dimension is F496, and the 17-dimensional distribution is around F200. On the circRNA versus IncRNA dataset, the first dimension feature is F404, and most are located between F200 and F300 F450. For the last dataset, the first dimension is characterized by F98 and F404 is the second dimension. Most of the feature distributions are the same as the second one. Moreover, we can get the common points of the 20-dimensional feature distribution on the three datasets. That is, most of the features are around F200 and F356. Finally, we analyzed which features are important for the identification of circRNA based on feature extraction. To analyze the distribution of local features, we divided the 520 features into four intervals: 1,148, 148,318, 318,364, and 364,520. According to the order and results of using the feature expression method. The distribution of the top 20 features in each interval is given in figure 4b. According to the top 20 features of the score and the interval map of the distribution, 4 falls into the interval 148,318, 7 into 318,364, and 9 into 364,520, in which the scores in the intervals 318,364 and 364,520 were also ranked higher. This shows that the structural features of RNA and pseudo-ribonucleic acid composition are most important, and the proportions of autocorrelation and ribonucleic acid composition are relatively less important. Therefore, we concluded that the structural features and pseudo-ribonucleic acid composition contain more feature information than the other two feature regions. 3.2. Optimization of kernel function of ELM. For ELM with excellent classification performance, it is important to choose an appropriate and stable hidden layer activation function. This section presents a discussion of the different classification performances of different activation functions and how to find the optimal activation function. For optimization of the kernel function, 5 common activation functions are selected here: sigmoid, sine, hardlime, tribase, and radial basis function, RBF, 60. 5 activation functions were used to the 3 datasets, respectively. The classification results are evaluated based on 4 indicators: ACC, SE, SP, and MCC. The recognition results were obtained under tenfold cross-validation, figure 5a. We found that the values of MCC, SP, and SE of RBF are not the best for the dataset of circRNAs expressed and not expressed in stem cells. However, the effect is better than the other 4 functions for the other 2 datasets. The results for the 3 datasets are as follows: accuracy of 0.784, 0.794, and 0.749, SE of 0.762, 0.679, and 0.749, and SP of 0.75, 0.839, and 0.739, respectively. In general, the RBF function achieves the best recognition of the overall effect and proves the validity of RBF for recognizing circRNA. Therefore, RBF was selected as the activation function of ELM. To further prove the stability of the classification effect, the activation function was verified on an independent test set. The results are shown in Figure 5b. According to the trend of the fold line, the RBF function is at a higher position for the three datasets in terms of accuracy, and the accuracy of the independent test set is higher than the tenfold cross-validation effect. This also proves that the recognition effect of RBF has certain stability and effectiveness. Therefore, in the following experiments, RBF is used as an activation function of the hidden layer. 3.3. Optimization of ELM using PSO algorithm. The traditional ELM lacks effective training methods, and it has the disadvantage of poor prediction accuracy, 61. In contrast, PSO, as an intelligent optimization algorithm, can improve the performance of ELM. Here, the PSO algorithm was used to optimize ELM, and it was compared with basic ELM results and the optimization effect of genetic algorithm, 62. The results of experiments of tenfold cross-validation show that, compared with the recognition results of the optimized ELM and the basic ELM algorithm, GAELM and CirRNAPL improved to some extent, Figure 5c. In terms of the classification effect, ELM and CirRNAPL achieved better results than GAELM. On the three datasets, CirRNAPL achieved accurate ACC values of 0.815, 0.822, and 0.782. Experiments showed that Figure 4. Feature importance analysis. A. Top 20-dimensional feature distribution on three datasets. B. Feature importance analysis. On the three datasets, 520-dimensional features were obtained by feature selection. The 520-dimensional feature distribution was organized. 838 MU et al. Computational and Structural Biotechnology Journal 18, 2020, 834 to 842. The PSO effectively improved the prediction accuracy and generalizability of the ELM network. Therefore, the improved ELM was used as a classification algorithm to identify circRNA. 3.4. Comparison with other classifiers. To verify the validity of the CirRNAPL, the classifier was compared to other classification algorithms. Based on Section 3.2 and Section 3.3, RBF is the selected activation function and the ELM was improved using the PSO algorithm. Next, the classification effect of the improved ELM algorithm was compared with those commonly used algorithms, such as CNN, convolutional neural networks, 63, RF, support vector machine, SVM, J48, 64, and ZERR, 65. The comparison results are shown in Figure 5d. As shown in Figure 5d, from the comparison of the results of ACC, SE, SP, and MCC, compared with the CNN, RF, SVM, J48, and ZERR algorithms, the classifier CirRNAPL constructed here achieved good results. For the three datasets, CirRNAPL achieves recognition accuracy of 0.815, 0.822, and 0.782, and also demonstrates the effectiveness of PSO ELM for identifying circRNA. After verifying the classifier CirRNAPL under tenfold cross-validation, to further explain the effect, CirRNAPL was further validated here on an independent test set. The results of the independent test set verification are shown in Figure 5e. According to the accuracy values of the 5 algorithms on the 3 datasets, the classifier CirRNAPL constructed in this study achieved a high accuracy rate. The accuracy rates for the 3 datasets are 0.887, 0.906, and 0.841, and the accuracy of the independent test set is higher than the accuracy of the tenfold cross-validation. The findings also prove that the classification accuracy of the tenfold cross-validated classifier is reliable and stable. 3.5. Comparison of the results with the traditional BLAST method. This section mainly describes a comparison of the recognition effects of the constructed classifier CirRNAPL and the traditional BLAST sequence alignment. In biological research, if a previously unknown gene is found, then a standard method, such as the base alignment search tool BLAST, is applied to obtain useful information through comparative analysis. With the development of machine learning algorithms and information technology, an increasing abundance of methods is becoming available for applying machine learning algorithms to identify unknown genes. To combine the effect of the traditional BLAST and the identification effect of the machine learning method, we compared the identification effect between CirRNAPL and BLAST. When performing BLAST, All sequence files are database files queried by BLAST, and each sequence is used as a query file in turn. Each sequence can get a BLAST result file, and the results are arranged in descending order of identity/length. Then, the prediction category of the query sequence is the category corresponding to the sequence with the largest identity/length value. The final result is shown in Figure 5F. We found that the classification accuracy of BLAST is clearly lower than that of CirRNAPL. The recognition accuracy of BLAST is 0.439, 0.605, and 0.611, while the classification accuracy of CirRNAPL is 0.815, 0.802, and 0.782, respectively. Given that BLAST only compares certain keywords that are more or less important in the sequence, it is not surprising that the accuracy is slightly lower. Thus, there is no doubt that the sequence database CirRNAPL classification methods will have increasingly broad validity and usability in research. 3.6. Comparison with state-of-the-art methods to test the effectiveness of the identification of the proposed classifier CirRNAPL. We compared its identification performance. Figure 5. Classifier validity verification. A. Identification results of 5 activation functions under tenfold cross-validation. B. Validation of the kernel function on the independent test set. C. Optimization of the experimental results of the extreme learning machine. D. Results of comparison with other classifiers under tenfold cross-validation. E. Validation of classifiers on independent test sets. F. Comparison of identification results compared with traditional BLAST sequences. Mu et al. Computational and Structural Biotechnology Journal 18, 2020, 834-842, 839 with that of existing classifiers, Web circRNA, circRNA, HELM, circDeep, Figure 6a. Among these models, the literature, 31, only used SE and SP indicators among the 4 commonly used evaluation indicators. circRNA, HELM, and circDeep only used the circRNA versus LungRNA dataset. Firstly, we compared the results of CirRNAPL with Web circRNA. From Figure 6a, we found that CirRNAPL achieved better performance on the stem cell versus NOT and circRNA versus PCG datasets than Web circRNA, and in contrast, the performance on circRNA versus LungRNA was slightly worse. We analyzed the causes of the performance on the circRNA versus LungRNA data set from the perspective of features. According to the score generated after feature selection, the paper selected the two-dimensional features that effect is better in the results of each feature expression method and used violin plots to show the data distribution on the positive and negative example data sets, Figure 6b. From the Figure 6b, we can see Figure 6. A. Comparison with state-of-the-art methods. B. Analysis of the importance of violin diagram features. Figure 7. A. Relationship between sequence alignment and E-value under stem and non-stem. B. Analysis of the relationship between the expression level and abundance of the sequence. C. Sequence alignment partial conservative region display and consensus log. 840 Mu et al. Computational and Structural Biotechnology Journal 18, 2020, 834-842 that the difference of feature distribution is relatively small between positive and negative examples, showing that the feature representation method we use cannot represent the characteristics of circRNA and LungRNA well. This result indicates that the feature representation method, which can make a significant difference in the distribution of positive and negative samples, would greatly contribute to the final classification result. It brings new challenges on how to use more efficient features and improve the performance of the classifier. Secondly, we compared the results of CirRNAPL with circRNA, HELM, and circDeep, respectively. From Figure 6a, we can know that the performance of CirRNAPL is better than circRNA and HELM in the 3 indicators of ACC, SE, and MCC, while lower than the latest circDeep. The ACC value of the circDeep method is higher than CirRNAPL. By comparison, we can know that although CirRNAPL has achieved some progress, more advanced techniques are required for better identification results in future research. 3.7. BLAST sequence alignment expression analysis based on RNA-seq data after the prediction of circular RNA. We further consider whether it is possible to analyze the expression of circular RNA based on the available dataset. We explore the relationship between the level of sequence expression and the abundance of the sequence, the number of reads on the alignment. So, we downloaded the RNA-seq data from the GEO database, https://www.ncb.nlm.nih.gov/geo, which is used for similarity detection. The independent test set of the third dataset in the paper was used as a query sequence and included 282 positive samples that were expressed as H1-seq in the stem cell and 282 negative samples that were not in the stem cell and were not expressed as H1-seq. Similarity sequence alignment was then performed by local blast, with different E-values, including 1.0, 0.1, 0.01, 1E3, and 1E4, respectively. For the two groups of 282 query sequences, we recorded the values of different sequence alignments under different E-values, figure 7a. When the E-value was at least 1E3, the numbers of sequence alignments on the two datasets were 64 and 69. In the case of the stem and no stem, the changing trend of E-value and expression level is the same. It can be seen that the influence. Then we calculated the abundance of each circRNA and then obtained a scatter plot of the relationship between expression and abundance, figure 7b. The scatter plot implicates that the abundance of the sequence is high when the expression level of the sequence is high. Then, the polynomial fitting of the obtained data points was carried out, and two fitting curves were obtained. According to the trend analysis of the fitted curve of figure 7b, the relationship between the expression level of circRNA and the sequence abundance is the same in both cases. The expression level increases with the increase of abundance. The correlation coefficients in the two cases were 0.840 and 0.899, respectively, and the correlation of not stem was higher. We can also see that there are more sequence abundances on the not stem data. When the expression reaches a certain level, it does not change even though the sequence abundance increases. It shows that the expression level and abundance of the sequence are positively correlated. After analyzing the expression level, we randomly selected 8 sequences for multiple sequence alignment. The multi-sequence alignment of the circRNA was obtained by the Clustal Omega online alignment tool, and some of the conserved regions were displayed, figure 7c. The results showed there are different colorations of nucleic acids in conserved regions while the consensus logo in the conservative region, 66. The nucleic acid distribution characteristics of the conserved regions of the circRNA were revealed by the displayed logo map. 4. Conclusion: we constructed a new classifier for the accuracy of identification of circRNA, and the effectiveness of the identification of circRNA has been demonstrated based on 3 publicly available datasets. Through the analysis of feature importance, we found that the structural features and pseudo-ribonucleic acid composition features showed better performance. However, the performance to distinguish circRNA and lncRNA is not very good, and it is still a widespread problem to distinguish them. More effective features and data are urgently required. Additionally, sequence alignments of circRNAs were preferably analyzed based on RNA-seq data. The comparison with the circDeep method also prompted us to use more efficient and updated techniques to improve the performance of our model. Planned future work includes not only finding more useful features but also using advanced parallel technology to identify the growing number of circRNA sequences. Both of them will improve the efficiency of identification. Meanwhile, given that the functions of most circRNAs are still unknown and little work has been performed on the large-scale discovery of disease-related circRNAs, future studies should focus on the biological significance of circRNAs. 5. Data accession numbers: the RNA-seq data is from the GEO database, https://www.ncb.nlm.nih.gov/geo. The GEO number is GSE 63823. Credit authorship contribution statement: Mengting Yu, writing, original draft, methodology. Zhang, conceptualization, software. Yanjuan Li, project administration. Cankun Wang, formal analysis, validation. Zhaoqian, writing, review and editing. Hui Ding, data curation. Quan, funding acquisition. Qin Mad, supervision. Acknowledgements: we thank all members in the ZO lab for helping in paper writing and server support. We also thank Marlene Merling for language help. Funding: the work was supported by the National Key R&D Program of China, 2018 YFC 09104005, and the Natural Science Foundation of China, number 61,771,331, 61,300,098. The work was supported by the Fundamental Research Funds for the Central Universities, China, No. 2572017 CB33. References: 1. Hao S., LVJ, Yang Q, Wang A, Li Z, Guo Y, et al. Identification of key genes and circular RNAs in human gastric cancer. J. MedSci Monitor 2019:25 to 2488 to 504. 2. Bogard B., Francastel C., Hube F. A new method for the identification of thousands of circular RNAs. J. Non-coding RNA Investigation 2018-2. 3. Diener T. Potato Spindle Tuber Virus, 4. A replicating, low molecular weight RNA. J. Viral 1971:45 to 411 to 28. 4. Cheng J., Mika F., Dietrich C. Specific identification and quantification of circular RNAs from sequencing data. J. Bioinformatics 2015:32 to 1094 to 6. 5. Dori M., Alia LHA, Cavalli D., Massolini S., Lesh M., Dahl A., et al. Sequence and expression levels of circular RNAs in progenitor cell types during mouse corticogenesis. J. Life Sci Alliance 2019:2:E201900354. 6. Song X, Zhang N., Han P., Moon B.S., Lai R.K., Wang K., et al. Circular RNA profile in gliomas revealed by identification tool Uroboros. J. Nucleic acids Res 2016:44:E87. 7. Li S., Han L. Circular RNAs as promising biomarkers in cancer, detection, function, and beyond. J. Genome Med. 2019:11:15. M. Yu et al. Computational and Structural Biotechnology Journal 18, 2020, 834-842, 841, 8, Glazar P., Papavasileu P., Rajewski and CircBase, a database for circular RNAs. J. RNA 2014:20 to 166670. 9. Lu YC, Li JR, Sun CH, Andrews E., Zhao RF, Lin FM, et al. CircNet, a database of circular RNAs derived from transcriptome sequencing data. J. Nucleic acids Res 2015:44:D20915. 10. Casal S., Das S., Sen R., Basak P., Chakrabarty J. Circ2 traits, a comprehensive database for circular RNA potentially associated with disease and traits. J. Front Gen 2013:4 to 283. 11. Xia S., Feng J., Lei L., Hu J., Xia L., Wang J., et al. Comprehensive characterization of tissue-specific circular RNAs in the human and mouse genomes. J. Briefings Bioinformatics 2016:18 to 984 to 92. 12. Chen X, Han P. Zhou T, Guo X, Song X, Li Y circRNAPL, a comprehensive database for human circular RNAs with protein coding annotations. J. CirREP 2016:6 to 34985. 13. Du Kula DB, Panda AC, Grammaticacus I, the S, Abdel Mohsen K, Barospe M. Circ Interactome, a web tool for exploring circular RNAs and their interacting proteins and microRNAs. J. RNA Bio 2016:13:34 to 42. 14. Gong X, Wu Ji, Sung Si Role of Circular RNAs in Cardiovascular Diseases. J. Experimental Bio Med 2019:244 to 73 to 82. 15. Qian M, Chen R, Li T, Xiaobi Reduced Expression of circRNA *circ_0003159 in gastric cancer and its clinical significance. J. Clin Lab Anal 2018:32:E22281. 16. Yao T, Chen Q, Xiao Z, Song Z, Fu L, Xiaobi Circular RNA 0068669 is a new biomarker for hepatocellular carcinoma metastasis. J. Clin Lab Anal 2018:32, E22572. 17. Huang J.T. Chen J.N., Gong L.P., Bai Y.H., Liang J. Zhou L. et al. Identification of virus-encoded circular RNA. J. Viral 2019 529 to 144 to 51. 18. Miao Q, Zhang Z, Lin Y, Ni B, Yang W, Tang J. RNA-seq of circular RNAs identified circ22 as a potential new activity indicator in systemic lupus erythematosus. J. Lupus, 2019. 09612033198304993. 19. Shu Y, Ren Y, Lin T. Hui D. Identification and characterization of circRNAs involved in the regulation of wheat root length. J. Biol Rees 2019 5219. 20. Yisai, Chen L, Lu C, Ju QH, Fan L. Widespread non-coding circular RNA-S in plants. J. New Phytol 2015 208 to 88 to 95. 21. Mumzak S, Jens M, Elefthaniode, Tordieff, Kruger J, Ryback A, et al. Circular RNAs are a large class of animal RNAs with regulatory potency. J. Nature 2013 495 to 333. 22. Hansen TB. Improved circRNA identification by combining prediction algorithms. J. Front Cell Devil Bio 2018 6:20. 23. Chen L, UY Zhang X, Lucy, UC, Fan Elk circRNA_finder, a software for circRNA prediction in plants. J. Bioinformatics 2016 32 to 3528 to 9. 24. Danon M, Schwartz S, Adelheit S, Sorkar transcriptome Y discovery of circular RNAs in archaea. J. Nucleic acids Rees 2011 40 to 3131 to 42. 25. Yek WR, Sharpless Ni. Detecting and characterizing circular RNAs. J. Nature Biotechnology 2014 32 to 453. 26. Gao Y, Wang J, Zhao F. Siri, an efficient and unbiased algorithm for de novo circular RNA identification. J. Genome Bio 2015 16 to 4. 27. Zhang XO, Wang HB, Zhang Y, Lu X, Chen LL, Yang L. Complementary sequence mediated exon circularization. J. Cell 2014 159 to 134 to 47. 28. Bo JN, Cheslik M, Zhang Y, Shukla S, Xiao L, Zhang Y, et al. The Landscape of Circular RNA in Cancer. J. Cell 2019 176 869 to 881, E813. 29. UX, Conrad 2. X. Accurate circRNA identification and quantification from RNA-seq data. J. CirREP 2016 6 to 38820. 30. Zhang QL, GXY, Li HW, Guo J, Wang F, Deng XY, et al. Identification of circular RNAs and their altered expression under poly-I-C challenge in key antiviral immune pathways in amphioxus. J. Fish Shellfish Immunal 2019 86 to 1053 to 7. 31. Pan X, Xiang K, Anthon C, Hiddle P, Freude K, Jensen L. et al. Web circRNA, classifying the circular RNA potential of coding and non-coding RNA. J. Genes 2018 9 to 536. 32. Pan X, Xiang K PreCircRNA, computational classification of circular RNA from other long non-coding RNA using hybrid features. J. Molecular Biosys 2015 11 to 2219 to 26. 33. Chen L, Zhang YH, Huang Ji, Pan X, Wang S, Huang T, et al. Discriminating cirRNAs from other circRNAs using a hierarchical extreme learning machine, HELM, algorithm with feature selection. J. Molecular Gen Genomics 2018 293 to 137 to 49. 34. Chaban M, Williams RM, Stevens at, Park JW. CircDeep, Deep Learning Approach for Circular RNA Classification from Other Long Non-Coding RNA. J. Bioinformatics 2020 36 73 to 80. 35. Wei L, Liao M, Gao X, Zhou Q. An improved protein structural classes prediction method by incorporating both sequence and structure information. JIEEE Trans-Banobayashi 2015 14 to 339 to 49. 36. Wei L, Sing P, Tang J, Zhaoqian Pred RF, a novel sequence-based predictor for phosphorylation sites using sequential information only. JIEEE Trans Nanobiology 2017 16 to 240 to 7. 37, UM, Li Y, Wang C, Han K. RF Amyloid, a web server for predicting amyloid proteins. J&J Molecular Sci 2018 19 to 2071. 38, Zhang L, Zhang J, Xuan P, Zhou Q. BP neural network could help improve PRMRNA identification in various species. J. Biomed Res in 2016 2016. 39, Dong R, Ma XK, Li GW, Yang L circPedia V2, an updated database for comprehensive circular RNA annotation and expression comparison. J. Genomics, Proteomics Bioinform 2018 16 to 226 to 33. 40, GP, Wu W, Chen S, Zheng Y, Zhou L, Zhang J, et al. Expanded Expression Landscape and Prioritization of Circular RNAs in Mammals. J. Cell Rep 2019 26, 3444 to 3460, E3445. 41, Lu B, Lu F, Wang X, Chen J, Fang L, Cho KC. Xin1, a web server for generating various modes of pseudo-components of DNA, RNA, and protein sequences. J. Nucleic Acids Res 2015 43 W6571. 42, Wei L, Liao M, Gao Y GR, He Z, Zhou improved and promising identification of human microRNAs by incorporating a high-quality negative set. JIEEE/ACM Trans Compute Bioinform, TCBB, 2014 11 to 192 to 201. 43, Luo L, Li D, Zhang W, 2S, Ju X, Qianji accurate prediction of transposon-derived PRMRNAs by integrating various sequential and physicochemical features. J. Plos 1 2016 11 E0153268. 44, Zhang W, UE predicting flexible length linear B-cell epitopes using pairwise sequence similarity. In 2010 Third International Conference on Biomedical Engineering and Informatics, Volume 6. IEEE, 2010. p. 2338-42. 45, Lodi H, Saunders C, Shaw Taylor J, Christianini N, Watkins C. Text classification using string kernels. J. Machine Learning Res 2002 2 to 419 to 44. 46, Dong Q, Zhou S, Quan J. A new taxonomy-based protein fold recognition approach based on autocross covariance transformation. J. Bioinformatics 2009 25 to 2655 to 62. 47, Friedel M, Nicola Jua S, Sunil J, Wilhelm T. D. Prod, a database for dinucleotide properties. J. Nucleic Acids Res 2008 37 D3740. 48, Chen W, Zhang X, Berker J, Lin H, Zhang L, Cho KC. SKNC General, a cross-platform package for generating various modes of pseudonucleotide compositions. J. Bioinformatics 2014 31 to 119 to 20. 49, Chen W, Feng PM, Lin H, Cho KC. ERSpots DNC, identify recombination spots with pseudo-dinucleotide composition. J. Nucleic Acids Res 2013 41 E68. 50, Lu B, Feng L, Lu F, Wang X, Chen J, Cho KC. Identification of real microRNA precursors with a pseudostructure status composition approach. J. Plos 1 2015 10 E01215001. 51, UZH, Laik, Ju L, Xia J, Wangd prediction of protein-protein interactions from amino acid sequences with ensemble extreme learning machines and principal component analysis. JBMC Bioinformatics 2013 14 S10. 52, Cao J, Lin Z, Huang GB, Lu N voting-based extreme learning machine. J. Inform Sci 2012 185 to 66 to 77. 53, Cao J, Xiang L protein sequence classification with improved extreme learning machine algorithms. J. Biomed Res in 2014 2014 to 1003054. 54. Wangd, Huang Protein sequence classification using extreme learning machine. In IEEE International Joint Conference on Neural Networks, 2005. IJSN 05. Proceedings. Volume 1403, 2005. p. 1406-11. 55. Huang Juqi, Su CK. Extreme Learning Machine, Theory and Applications. J. Neurocomputing 2006 70 to 489 to 501. 56. FOM-D, Karaboga D. Intelligent Optimization Techniques, Genetic Algorithms, to Boost Search, Simulated Annealing in Neural Networks. Springer Science and Business Media, 2012. 57. Li Y. U. M. Huo J. An Inductive Logic Programming Algorithm Based on Artificial Bee Colony. J. Inform Technol Res, JITR, 2019 12:89 to 104. 58. BayQ Analysis of Particle Swarm Optimization Algorithm. J. Comp Inform Sci 2010 3 to 180. 59. Zhang J. Cao L. Gr. A novel features ranking metric with application to scalable visual and bioinformatics data classification. J. Neurocomputing 2016 173 to 346 to 54. 60. Javed K. Goraev O.R. Zerhouni N. SWELM, a summation wavelet extreme learning machine algorithm with a priori parameter initialization. J. Neurocomputing 2014 123 to 299 to 307. 61. Wang Y. Cao F. Yuan Y. A study on effectiveness of extreme learning machine. J. Neurocomputing 2011 74 to 2483 to 90. 62. Deb K. Pratap A. Agarwal S. Mayrivan T. A fast and elitist multi-objective genetic algorithm, NSGA2. JIEEE Trans Evolution Compute 2002 6 to 182 to 97. 63. Fianica A. La Rosa M. La Palia L. Rizzo R. Urso A. NRC, non-coding RNA classifier based on structural features. J. Biodata Mining 2017 10:27. 64. Patil Tr. Sharikar S. Performance Analysis of Naive Bayes and J48 Classification Algorithm for Data Classification. J and J Comp Sci Opel 2013 6 to 256 to 61. 65. Aher SB, Lobo Al Comparative Study of Classification Algorithms. J and J Inform Technol 2012 5 to 239 to 43. 66. Waterhouse AM, Proctor JB, Martin DM, Clamp M, Barton GJ. JAVU version 2, a multiple sequence alignment editor and analysis workbench. J Bioinformatics 2009 25 to 1189 to 91. 842 MU et al. Computational and Structural Biotechnology Journal 18, 2020, 834 to 842